by the ITTO and BMLEH Teak projects. So far we have done a very good job. And today the fifth one, and we still have two more before the end of the project in December. So the webinar theme today is the Teak DNA barcoding and forensic investigation for legal supply chain. So as you know, right now for the international timber trading, there are a lot of the mechanism for traceability and checking the certification. So DNA become a promising techniques because the analog technique or the paperwork. So before we start, I briefly discuss again. So this webinar is supported by the ITTO BMLEH project phase 2, funded by the Federal Ministry of Agriculture, Food and Regional Identity from Germany. So we have 6 participating countries, so 5 countries in Asia and Pacific, namely Cambodia, Thailand, Vietnam, India, and Indonesia. And we have Togo in west of Africa. So the project started in 2019, the project phase 1. So focus on the upstream activities. And this one is the project phase 2. We started in 2023 and will be ended in coming December 2026. We focus on activity in the intermediate stream. So today the DNA barcoding and forest certification is highly related to the project. Okay. So I show you in the PowerPoint, you see a lot of webinar have been conducted so far. So today is the 10th webinar, and we have 2 female speakers, very knowledgeable people. So before we actually start the webinar, so may I invite Jennifer Conte, Director of Forest Management Division of International Tropical Timber Organization, to deliver welcome remark on behalf of ITTO. Jennifer, please.
Thank you, Dr. Youngert. Can you hear me okay? Yes?
Yes, okay. We can hear you very well.
Okay, wonderful. So on behalf of ITTO, I'd like to extend warm regards and sincere appreciation to all the participants today and our distinguished speakers and all the engaged academics and practitioners of forestry attending today's webinar. As Dr. Youngert has already explained, this is part of the ITTO BMLEH Teak project. And the aim of these webinars are to strengthen regional and international collaboration, information sharing, knowledge management, and uptake on the management of smallholder teak and other valuable species plantations. Today's theme is teak DNA barcoding and forensic investigation for legal supply chains. So in addition to poor quality planting stock, inadequate silvicultural practices, and weak marketing and value chains, the verification and traceability of timber species and geographic origin still remain major challenges to establishing legal timber supply chains, particularly in international trade. Many of you are already aware of the technologies that exist out there, but just to give a quick overview, DNA barcoding is a molecular identification technique that uses a short standardized region of DNA to identify a species, much like you would use a supermarket barcode to identify a product. Timber forensics applies molecular genetics, chemistry, and wood anatomy to determine the species, geographic origin, and legality of timber products. And together, these approaches provide scientific evidence to verify claims made throughout the timber supply chains. These technologies are used to strengthen due diligence systems by verifying the species and origin declared in harvest permits and trade documents. It's used to support the enforcement of regulations such as the EUDR, the US Lacey Act, the Australian Illegal Logging Prohibition Act, and for FSC and PFC certification schemes. It also provides scientific evidence for investigations into illegal logging and timber laundering. So in today's webinar, we are privileged to learn from two distinguished scientists. Both are accomplished female researchers with extensive experience in forest genetics and tree breeding. So please join me in welcoming our guest speakers. The first is Dr. Suma Arun Dev, who is a senior principal scientist in the Kerala Forest Research Institute in Kerala, India. She is a forest geneticist whose expertise includes forest genetics, tree breeding, molecular biology, DNA barcoding for species identification and biosystematics, and conservation genomics of several important tropical tree species, including teak, sandalwood, bamboo, rattan, and other endemic forest species of the Western Ghats of India. Today, she'll be presenting on forensic tools for tracing timber theft and geographic origin in legal timber supply chain. Our second speaker is Professor Ulfa Siregar, who is with the Faculty of Forestry at IPB University in Indonesia. Professor Ulfa Siregar's expertise and research focuses on forest genetics, silviculture, provenance trials, and the conservation and breeding of important tree species, including Gelatung, Acacia magnum, teak, and microalgae. And she will be speaking today on DNA barcoding for valuable tree species in Indonesia. So on behalf of ITTO, I'd like to express our sincere appreciation to these two speakers for sharing their expertise on these emerging topics, which are critical to strengthening legal and sustainable timber supply chains. And last but not least, I'd like to extend my sincere thanks to Dr. Yuanyardi, Tetra Yuanyardi, who is the ITTO project manager, Dr. Juan Okma, and Professor Yongart Prisuart for organizing this webinar. And as always, we are very grateful to the donor, Germany, for their continued support, and to Kasetsart University and the 6 participating countries, as well as TICNA. I wish you all a very successful webinar and hope it proves insightful and fruitful for all of you today. Thank you so much.
So thank you very much, Jennifer, for your welcome remark for today. So as you already said, I think that today's topic is very interesting and is highly required for testability. So I have opportunity to see the slide of both speaker. So with your permission, so may I start with Professor Ufa from Indonesia, because the presentation is highly related to theory, while Dr. Suma is related to the application. Okay? Thank you. So without further ado, I will start with the presentation of just a moment. Okay.
Okay. Thank you very much, Professor Yang Trisurat.
Okay.
So,
I upload your.
no, can I share
Okay.
my screen?
You can share your presentation from from your
okay.
Okay, that's good.
Can everybody see my
Yeah, we see your
slides?
PowerPoint now. Go ahead. Please.
Okay. Okay. I will put it in slideshow. Okay. Okay, I'll start. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Yeah, I got invitation from Professor Yang to present these topics, DNA barcoding for valuable tree species in Indonesia. Especially this is for forensic purposes, something like that. So in my presentation, I will share our experience a little bit. Next. So as you probably know, so Indonesia has many problems with this illegal timber. And yeah, monitoring as shown here, reveal Indonesia illegal timber seed. Most of the illegal timber came from document falsification. So this you can see here. And there is misdeclaration of timber origin or species. And then custom fraud, of course. And then there is what we call logging outside concessions boundaries. So this mainly the source of this illegal timber. And as reported by WRI in 2026 recently, in 2013, Indonesia was among the world's 3 largest producers of illegal timber. Approximately 50% of the timber production estimated to originate from illegal sources. That's a lot. Luckily, Indonesia is the only country, according to WRI, authorized to issue black license and has implemented what we call SVLK, our timber legality assurance system, through mandatory certificate. And if this illegal timber is caught, they will be prosecuted under the forestry law number 41, 1999, and the law on the prevention and eradication of forest destruction, law number 18, 2013. So I will give you just a little bit outline of this timber legality system. Yes, timber legality system in Indonesia. So this legality system is, of course, developed by government. And then this VB accreditation by CAN. CAN is accreditation, national accreditation. And then from this one, and then this already passed and was distributed among forest management unit and industries, forest industries, something like that. Just this is just how this SVLK was developed and now established and applied. Now, that is the introduction part, and I will come to my main topic of my presentation. Of course, if the illegal timber, you cannot detect by document because document can be falsified, like previously in my slide. We need to certain method which can guarantee that we can detect the illegal timber so that we can bring it to justice, something like that. And molecular good identification, provide that good identification technologies. So now I'm talking about the molecular method, supports law enforcement because provide scientific evidence as recognized by under Supreme Court. This is in Indonesia, number 1, 2023. And limit under Article 39, Forest Destruction Prevention, PDH law, we call it, and then enable prosecutors to pursue additional or criminal sanctions, and also forestry non-tax state revenues referred to the SDH. This is Indonesia, so something like that. So we can apply, we can use molecular root identification to prove whether this timber is legal or illegal. And in the good identification, we know there are 3 types of identification method I could call it method or technology. That is, number one is conventional DNA markers like RFID, microsatellites, SSR, IFLP, and so on. And then second is this what is supposed to be my talk today is DNA barcoding. And then maybe this is most recent one is single nucleotide polymorphism or SN, which is abundant and more specific markers that we can find in the genome of certain species. As you can see here, so using this molecular wood identification, actually we need to identify the species. We should identify the population from where the wood is coming from. Yeah, so this is the population genetics, and of course, if possible, because the DNA barcoding is identify the species, so we should go deeper. If possible, we should identify the individual profiling or fingerprinting, something like that. So there are three challenges that molecular wood identification need to do to be able to support law enforcement in bringing the illegal timber supplier to justice, of course. So this is about DNA barcoding. I think everybody knows DNA barcoding. Yeah, DNA barcoding is using certain sequences, short sequences, to identify a certain species and, if possible, certain population of the timber or the wood. And you can see here core sequence for DNA barcoding is the length should be short, between 400 to 800, not too long. And then about variation, this sequence should generate high divergence among different species. And then the conservation part is it should be low variation within the same species. So interspecies variation should be low. So this is becoming the limitation of DNA barcode. Later I will explain why. And universality. So this sequence should be flanked by conserved region so that primer can attach to this DNA sequence, and we can amplify target across broad taxonomic groups. And the quality should be good, high signal to noise ratio with minimal ambiguity, insertion, deletion, or PCR levels. As you can see here, usually the DNA barcoding use genes or sequencing from genes in the chloroplast genome, mostly like MATK, RBCL, LPOP, and so on here. And sometimes we also use sequencing from sequences from nuclear genes, ITS or ITS2. And this is the application of cpDNA, cloroplast DNA, or DNA barcode for various. For example, this is for taxonomic studies when you identify certain species. So it's for taxonomy and then biodiversity assessment, organism, plant breeding, and here for forensic plant as well. So the problem, as I mentioned earlier, is this one. So it should have low variation in intraspecific variation. Low variation in the intra. Why? Because the DNA barcode actually is meant for differentiating species. So in order to be able to do that, you should not has variable, so high variable within the species. Otherwise, you fail. Then there is certain species, something like that. And this is becoming the limitation of the DNA barcoding for forensic purpose. Because in forensic purpose, it's like your fingerprint. It should be individually specific. So if low intraspecific, that means individually you almost cannot, although you still can differentiate individuals within certain species or something like that. So this is becoming the limitation, but it's still possible to do forensic using DNA bank. And here I want to show some of our experience in doing some research about using this molecular identification for forensic. So in Kalimantan, we try to how to say, to tag Dipterocarp species, and we investigate 2 companies, and we collect sample from the stump left in the field, and then a log file. So that means the log file after the log are cut and then filed in certain area, and then the log will send to wood industry, and then we also try to, this is Kalimantan Island, and then we try to trace back the illegal wood in Jakarta, something like that. So there are 2 companies similar. And what we found, we assess using microsatellite markers. And then after analyzing, we, as you can see here, the A stump here, and log file in company A. So this belong to company A. This is belong to company A. This is belong to company B, but this one is question. We don't know from where, something like that. As we can see here, the stump and the log and entry is all clustered into one cluster. That means wood from industry really coming from the logs file belong to company A, and must be coming from the stump, similar, that's a high similarity with the stump in the company. Company B also some similar. The log from industry is very related to log file in the company B, and also very related, closely related to the stump in the company. So that means this one is coming from the same source, this wood. And what is fascinating is the illegal logs closely related to the company. So that means the illegal logs we found in different islands, already sent to different islands, actually coming from the company, something like that. So this is and we also made some example using Teak, using RAP3, and that is similar. There is forest management unit C and P, and same, we take sample from the stump by log, and then the industry, and there is illegal, we don't know from. And as you can see here, P stump and P log and P logs file is same, very closely related. Stump and log file from C also closely related, and then wood industry, C wood industry, of course closely related with the C from company of C. So that means this, because this is C, so we know, and the illegal wood is coming from this one, not from this one, something like that. So both studies in the Dipterocarp and the Teak that we experience, look at it, similar, give similar result. So that is just using usual conventional molecular marker. And now there are many most advanced techniques which we can fingerprint individually or profiling. This method is rely on high throughput technology powered by next generation sequencing, which bypass traditional gel-based limitation to provide dense genome-wide single nucleotide or SNP profile. And there are several types here. This is technology, for example, GPS, RADSEq, and then KSP, DNA medical arrays, and so on. So I'm going to give you some example of what we were doing. It's not really for forensic, but we try to find SNP profiles for certain species. In this case, sengon, for 3 is pest and disease. So in our breeding program, we try to select trees resistant to certain pest and disease, and we try to genotype or fingerprint these 3 species, something like that. And we are using the transcriptomic and genomics approach, omics approach. So this is typical analysis using that. I'm not going to explain very detailed in this. And usually we will find the top 10 upregulated and top 10 downregulated genes. And from this, we know that by comparing this one, we know if 3 are susceptible to something or 3 is susceptible to something. There is different genes expressed, something like that, using this transcriptomic. And we detect, finally detect SNP. And we detect SNP using, in our case, we use HRR, the high resolution mapping, something like that. And we can see that there is difference in someone is leaking. Someone is leaking. Yeah. Okay. So from this result, we can. A to T, different base pair. Different base pair will result in different resistance. Similar here, from here to here, for example, from C to A, this becoming severely susceptible, something like that, and so on.
ทำไมเสียงไม่ออกอ่ะ
Sorry. I'm sorry, I will continue just a few more minutes.
ปิดนะปิดรูปนะปิดรูปนะปิดรูป
Somebody is talking.
แต่คำภาษาไทยเพื่อจะอ่านออก
รถตำรวจ
Sujat
สะกดด้วย
Kalyawongsa. Sujat Kalyawongsa. You are talking.
LICEF
Please mute microphone, please.
police
รถ
รถตำรวจ ROO
Anyone turn on microphone ใครเปิดไมโครโฟนช่วยปิดหน่อยครับ
Our tick, we
okay
อะไรของมัน
AIL
ครับ
please
ช่วยปิดไมโครโฟนหน่อยครับ
host can mute the unmuted participant. Please unmute the participant. Okay. Yeah. Okay. I will continue. Okay. So recently we start to study teak because in Indonesia, the teak plantation try to move into clonal plantation, something like that. And it's because one of our state company has developed what we call it fast-growing teak, and we try to compare the fast-growing teak with the conventional teak. As you can see, 20 years fast-growing teak and 70 years conventional teak, almost similar, something like that. So, of course, this is advantage to the state company, teak state company, and this one. Further results show that differences in good properties between fast-growing teak plant. It make it possible and easier to print the clothes using most advanced technology that I described, that I explained earlier. So from all of our research, this is lesson learned. Molecular genetic markers show consistency in timber forest product throughout timber chain of Kasetsart. So from the stump and then the log pile and then until the industry, all we can detect using molecular marker and show consistency. Of course, success in DNA isolation from wood is a precondition for this technology to be applied for timber, forensic timber. And requirement for full application of molecular markers and fingerprinting in forensic genetic in forestry depend on establishment of complete database. Yeah, we need to have database of genetic variation of all species that are commercially available, something like that, and the population. We need to establish the database, and of course, cheap and easy methods for DNA analysis. So this is just example of our publication for that research. And of course, that is not
Okay, finish. Dr. Professor Srika. Professor Srika, you hear me? Okay, thank you very much for your informative presentation.
that's all.
Yeah, thank you very much
Okay,
for your
that's
informative presentation.
You're welcome.
I'm sorry for the noise disturbance during your presentation. And congratulations for
Okay.
a lot of
No,
international and remarkable publication in the international journal.
yeah.
So I have
I'm sorry
limited,
that maybe the signal is not very stable from my side, maybe.
okay. So I have limited knowledge in the DNA. So may I invite the audience to comment and make presentation first? Any hand or comment or question? So we have Dr. Thani, Dr. Benjarat, who are expert in DNA. So would you like to ask question or comment?
Thank you very much.
Okay, Dr.
I'm
Thani,
Thani.
please. Yeah.
Hello. Can
Yes,
you hear
yes,
me?
we hear you. Go ahead.
Hello.
Yes, yes.
Yeah. Okay.
So thank you very much for your presentation. I'm very interested in your presentation. So actually, I want to know that to extract DNA from wood, it's quite difficult sometimes. For your experience,
Yes.
is it difficult to extract DNA from Teak? Because I have never tried DNA extraction from the wood tissue from the Teak.
Yes.
Thank you very much.
Several articles on how to extract DNA from wood. Yes, we try, and yeah, we somehow succeed. It depends on the wood, of course, and the extraction methodology. Sometimes we have to extract several times until we get the good data. But of course, we expect that the DNA from wood is not as complete as DNA from fish, for example. And we just try to apply in a PCR.
Yeah.
Okay, I check more hand.
Okay, thank you so much.
Yeah. So if no more hand, I have the general question, Professor.
Yes.
So I think I have no doubt or any question related to the technology of the DNA, because right now we have a lot of S1 technology to assess the DNA. But for the practitioner and administrator, usually they would ask what is the cost effective, okay, if we use the DNA barcoding for the forensic investigation compared to the traditional measure. So in practice, is that feasible for the general implementation?
For general implementation, it's probably similar like any other kind of cases in Indonesia. For example, some of our friends try to catch also many person who cause forest fire, for example, like that. And in such case, the police and the prosecutor, something like that, they will ask what do you call it, expert judgment, something like that, and then fact, they will ask us, and then we will investigate, and we will use it technology. Of course, land cannot do that. So inside, usually, police and prosecutor working together with expert, and then to prove, whether this illegal timber coming from Indonesia or this forest fire is due to somebody setting fire, something like that.
Okay. So it simply say that, so in general, so you use the traditional techniques. So just in case the police or the court would like to investigate in the detail for the case, right? Okay. Thank you.
So because this is very big and much, something like that, and then maybe police and prosecutor will try to catch the suspect or something like that, then they need to give evidence. And that's in such case, then the expert come in and helping them. Yeah.
Okay. Okay. So Kun Sapon, I see your hand. Kun Sapon, please.
Thank you, Professor Yongyut. From the presentation, I saw that you said that the cost of this technique is cheap. So in the former time, maybe 20 years ago, I have tried this DNA technique, but at the time very expensive. So I just wonder that at present time, how much does it cost per one sample if you want uh, to screen for the DNA analysis? Just in dollar or in rupiah is okay.
If it is just barcoding in Indonesia, let me think. Maybe one if only based on PCR, that is not so expensive. The most expensive is establishing the data first. So for example, you try to find the SNP, identify single nuclear. The expensive one is you try to sequence the whole genome. And then you try to sequence transcriptomic or something like that to find a certain marker. That once you already have a marker, you identify a marker, then you can develop a primer, and then just use usual PCR. And if usual PCR, something like that, you already get the primer. That is not so expensive. So the most expensive is the establishment first.
So in case if we already have everything and just for PCR, how much for one PCR for one sample?
For one sample in PCR in Indonesia, for example, rapid maybe around $10, $10, yeah,
Uh-huh.
one sample. Yeah, not so expensive. Yeah, not so expensive.
Uh-huh. Okay.
I think,
I just
yeah,
wonder
not
that
so expensive.
whether it, yeah, the technique is good. I just wonder that it is reasonable for using for the legal checks.
Okay, thank
It's
you.
already, if you
Thank you.
already have
Thank
the
you very much.
marker, yeah, just PCR, it's
Yeah.
not expensive.
Okay.
Okay.
Thank you. Thank you. So, Dr. Benjaras, how much does it cost for Thailand? Dr. Benjaras? No. Okay. Okay. Any more hand for the last question? So no more question. Okay. Thank you very much, Dr. Srika. Very informative presentation. Thank you very much.
You're welcome. Thank you.
So I hope we can see you in coming October in Yogyakarta. So may I proceed to our second speaker? So next second speaker is Dr. Sumar from India. So may I open up? Dr. Suma, you want manage your PowerPoint by yourself, or you want me to manage your PowerPoint?
No, we will upload from KFRA.
So you will manage your PowerPoint, right?
Yeah, I'll share from KFRA.
Okay. So you can share your PowerPoint from your side. Go ahead, please. Okay, we see your PowerPoint now, Dr. Suma.
Okay. Yeah, good afternoon, Professor Yongut and everyone in the webinar. At the outset, I thank Professor Yongut, the Regional Project Manager, ITTO BMLEH project, for providing me this opportunity to speak on forensic tools for tracing timber theft and for geographic origin in teak supply chain. Yeah, so just a minute. Yeah. So when it comes to the global teak resources, like as per the latest assessment of global teak resources, that is by the UFRO network group of scientists, as well as by the TeakNet, including Dr. Sandeep Collard and Sri Lakshmi. So it seems like the teak grows in 80 countries in the tropical regions. That is the most latest update of teak. And like the total area of the national teak bearing forest in the globally, it is 30.215 million hectares. So majority of these teak forests are in the tropical countries. I think half of it's in Myanmar, followed by India, Laos, and other tropical countries. So, and when it comes to the planted teak forest, it is coming to 4.854 million hectares. Of which 80% in Asia. So the three teak heavyweights, like when it comes to the planted teak forest, are India, followed by 35% of the planted forest are in India. It is followed by Indonesia and Myanmar. So we could make out that India remains the dominant trading power in teak, roundwood and salt timber market. And when it comes to the global timber trade, 97% of the teak timber that is being traded across the countries is coming to India from 43 source countries. So India always remains the dominant trading power when it comes to the teak timbers that are imported from different countries, tropical countries. So even though India remains the most, the dominant trading power when it comes to the teak timbers, at the same time, India is one of the favorite destination for the illegally traded timbers across the globe. So you can see that India is the third largest importer of the illegally logged wood after China and Vietnam. So this is because we don't have any stringent regulations in place when it comes to the
Sailesh, good afternoon. I was busy, so I did not see your message then. Tell me, Sailesh. You know how difficult it is to get a paper signed from our director. No?
So when it comes to the cross-border smuggling across the countries, like Myanmar is facing currently because of the complete ban on the export of logs as well as the logging. The Myanmar faces, it is one of the pressing issue in Myanmar, the illegal trading and smuggling of the teak timbers from its natural forest. So since India shares border with Myanmar in the Manipur state, most of these smuggled goods are coming to Manipur. So Manipur has become the hotspot for the illegally traded timber from Myanmar, and it is one of, again, it is from Manipur. It is being exported to globally to US and European Union countries, maybe like possibly as not as Burmese teak but as Indian teak. So that much rampant is the illegal cross-border illegal smuggling of timber from Myanmar to India. And even like we can make out that India shares like through Meghalaya, through West Bengal, a lot of illegally smuggled goods are coming to the country, like because India shares from Bangladesh, it is coming through Meghalaya and through West Bengal states, because in both the states, India shares borders with Bangladesh. So illegal timber trading or illegal cross-border timber trading is one of the serious issue. Even in our country, like we don't have any stringent means by which we will be able to distinguish the legally traded wood and illegally traded wood. So there requires some amount of some stringent regulations in place in the national level, so that we would be able to curb combat illegal felling as well as the cross-border transport of this illegally smuggled good. So when it comes to, even though we don't have any nationwide statistics when it comes to the illegal felling that is happening within the country, we can see, like as per one of the latest MOEF reports, like over the last 3 years, like 11 lakh trees were felled across 17 states in the country. And when it comes to the state records also, we can see like the illegal felling of teak trees are quite rampant in the country. And you can see maximum amount of illegal felling is happening in Maharashtra, and then the central India, and in Kerala also we had a very massive smelling of very recently a massive felling incident of teak trees. So within the country also illegal felling of the teak trees are happening, as well as we are facing cross-border smuggling of illegally felled trees from other countries. So this all highlights the relevance of legality, verification, traceability, and chain of custody systems in teak supply chain. So coming to the drivers of this illegal timber harvest and trade, as we know, like in India, like even though it is one of the major dominant power when it comes to the teak timber trade, and we have like when it comes to the highly priced timber tree species in the country, like we are having a huge domestic demand, and so this always there is a disparity or imbalance between the demand and supply. So this is one of the reasons, one of the drivers for the illegal timber harvest. And also, like we know, like there is a heightened focus of conservation. So the decline in the domestic production of wood in the country is because of the heightened focus of conservation in terms of Forest Conservation Act 1980, National Forest Policy 1988, and there is a complete ban of green felling in the country that is imposed by Supreme Court of India in 1996. As a result, we don't have that many resources, that many legal felling, because most of the forests, natural teak-bearing forests, are reserved. So we really face a scarcity of the timber. So there is an imbalance of demand and supply of teak timber in the country, which is of high demand in the country. So that is one of the key drivers for the illegal timber harvest. So illegal timber offense, actually, it's a very massive organized crime. So see, because of the very relaxed, or because of the absence of the stringent regulations, and this is actually affecting the environment, and also this is undermining the teak timber trade within the country. So, and as we know, like in many of the countries, very stringent rules are in place. And we know, like as Professor Ulfa was mentioning about the Lacey Act in the US, which is being implemented in 2010, and European Union Timber Regulations in 2010, as well as the Illegal Logging Prohibition Act in Australia 2012, and Cleanwood Act in 2017 in Japan. All these acts actually prevents the entry of illegally logged wood into their country. So all these developed countries are very stringent regulations in place. But in India, like we know, like there is no effective law enforcement, and we are just struggling to enforce the laws, because a country like where we have the limited means to implement laws, many of these crimes, many of the illegal felling or other timber theft offenses are cannot be dictated, because we don't have any clear-cut evidence, so that many of these convicts escape or illegal traders escape the conviction process. So, and then we know, like the identification of the timbers is being traditionally practiced by using anatomical means and by other means like organoleptic and other means and chemical means also. So most of these techniques which were currently in practice are not accurate enough for individualization of the timber when it comes to the supply chain. So, and when it comes to the illegal felling also. So we face an extreme difficulty when it comes to trace the origin of the timber. And this is actually an escape for the convicts, because they before the court, there is no scientific evidence to say that, okay, this timber is coming from this particular forest. So we don't have any measures by which you can look into the movement of the timber starting from the point of harvest through the supply chain till it reaches the consumers. So that is why the illegal timber felling is a serious offense in the country, and there are no means to curb the illegal felling that is happening in the country. So one reason is the huge domestic demand. Second is the heightened focus of conservation. Other one is the absence of effective law enforcement in the country. So the situation demands an adoption of integrated forensic approach involving both DNA tools as well as chemical tools. So this is being recommended by UNODC and also like a global timber tracking network. So globally, like almost all the international agencies have recommended the adoption of this integrated forensic approach to track the timber through the supply chain till it reaches the consumer. So the most important driver is in the country is the lack of robust forest certification. So when the teak timber through the supply chain, when it is coming into the market, we don't know like whether it is coming from a certified source, because as we know, like the forest certification is actually a multi-layer audit system that can authenticate origin, legality, and sustainability of forest-based product. So generally, certification is done to avoid consumption of any product that is the result of illegal logging. But these are the two international agencies, like we know globally, these are the two international agencies like Forest Stewardship Council as well as the PEFC. These two are the global certification agencies. So when it comes to India, like even the Forest Stewardship Council, it is been implemented in 2022 February, but it is still its infancy in the country. So it is not being taken up that far, because only Maharashtra, like 41 forest divisions in the state of Maharashtra is been only certified, and Uttarakhand also to some extent. So there is a lack of forest certification. We don't have any certified source of forest from which the teak timber that is coming into market. So recently the Ministry of Environment and Forest, Government of India, so just to gear up the NDA certification process, they have introduced an Indian Wooden Certification Scheme, in which we have like a third party, I mean a Praman, that is one of the very recently introduced scheme by Government of India under Indian certification scheme. So this will look into three types of certification. One is the forest management certification, because just to see like whether these woods are coming from certified and sustainably managed forest. Next is the trees outside forest certification, because we know like we don't have that, because there is a heightened focus of conservation. Most of the forests are reserved, and most of the domestic demand is met from the trees outside forest. So that has to be certified. So this is to monitor whether the wood from trees outside forest coming to market is from a certified source. And the last one is a chain of custody certification. So in this case, chain of custody certification will certify a timber when at the point of harvest through different steps in the supply chain till it reaches the point of sale or to the consumer. So that is, so all these three certification process are envisaged in the Praman scheme, which is very recently introduced by Government of India under MOEF, and Indian Institute of Management, Bhopal is the scheme operating agency. So, and then in addition to this, just to enhance the process of certification, there is another scheme that is introduced by Government of India, is the Vriksh scheme. That is for like the handicraft products. Whenever the handicraft products are made out of timbers, the Export Promotion Council has introduced this scheme for the timber legality assessment verification. So only the certified source through the Vriksh can be used for the manufacture of for the production of handicrafts. So all these, these are the key drivers why there is illegal felling is happening in the country, because we don't have any stringent regulations, stringent legal mechanisms in place, and also we don't have any certified forest. It is only getting certified maybe like another 20-30 years, like some of the forest resources may be certified under this Praman scheme. So we would be able to track the movement of the timber across the supply chain. So like in this context, I would like to emphasize the need for developing an advanced integrated timber-proof tool for timber traceability. So this tool involves both DNA tools as well as the chemical tools, and it is also recommended by United Nations Office on Drugs and Crimes, UNODC's best practice guide for forensic applications, and also by GTTN data analysis guide. So like we know, like there should be an adoption of an effective geographical signature, which can actually act as a certification standard for timber traceability. So coming to the DNA tools that can be used for forensic application, as Professor Ulfa was mentioning about DNA barcode. DNA barcode is can be very well used for species authentication. So whenever a timber theft is happening, DNA barcodes are developed from the DNA and say small conserved region. So that can serve as a biological barcode for a species. So we see when it comes to the smuggled good, when a timber theft offense is happening, when the seized logs are coming for identification, we don't have any taxonomical means by which you can confirm the authenticity. And wood anatomy has been practiced such a long time back itself, but in some instances it can identify only up to the genus level. So in this case, like if you employ DNA barcode, if you develop a DNA barcode database, then there is 100% accuracy for the species authentication. The next tool that can be used for forensic application that is already being used is the DNA profile for individual verification. So this has actually the DNA profiling or the DNA fingerprinting, just like we employ for paternity testing. It has mainly two applications. One is like whenever there is a illegal timber felling is happening, and if you need to identify the origin of the illegally felled timber, like where you can do the log to stump comparison. So in this case, this particular genome-wide tools can be used. And second is the individual assignment, like when it comes to the origin of the timbers, I mean the chain of custody validation, when it in the Teak supply chain, if you want to track the timber through the timber supply chain, you can use genome-wide markers, which has the ability for individualization of timber. So these tools are currently being practiced both for log to stump comparison in illegal timber felling, as well as for timber traceability and origin verification in Teak supply chain. So along with these tools, like when you do the assignment, like there are machine learning algorithms that's been developed in our lab, and like we are currently employing this machine learning algorithm. When it comes to the DNA barcode, like if you want to identify, if you have a reference database, say some 60 or 100 tree species, you already have the reference database, and if you want to identify one particular specimen that has come to you. What you like, it must be a seized log, and if you want to ensure the species authenticity, you will be developing the DNA barcode, and you will be having the DNA barcode sequence, and you will be testing it against the DNA barcode reference database which you have developed. So in that case, like in some instances, because if your database is so huge, you may not be able to visually identify. So in that case, you can use the VCA, that is a machine learning algorithm for barcode identification, you employing a database. And other two are assign, pop, and mycorrhiza. These are machine learning algorithms which is used for assignment of individuals when it comes to the geographical origin. So both these tools, both these machine learning algorithms can be used if you have already a database in place. That means, like, if you have developed, say for Teak, if you have a database of genome-wide tools in Teak, if it is within a country across different populations, you have to sample, you have to extract DNA, you have to develop your genome-wide tool marker, and then if you develop a DNA barcode database like in a country, and if you want to identify a particular timber, the origin of the particular teak timber in the teak supply chain, you can in fact use these two tools to see like this timber is originated from which population. So that is the application of these two tools. One is in the R package, one is a freeware, which is a Python-based, mycorrhiza is a Python-based freeware. So over the last 15 years, through various projects, KFRA has developed reference databases in many highly prized timber tree species, including teak. And so in the timber forensics, like actually what we do is like we use advanced scientific methods, say it is a DNA tool or a chemical marker, to identify the species. And the next is to identify the origin of the timber, in which we are to use a marker which has the ability for individualization of timber. So this can be used in the case of illegal logging operation, where you are to do the log to stem comparison. You can use it for timber theft, like when you want to ensure the authenticity of the species, and also for identifying the geographic origin. So when it comes to Teak, this particular work we initiated in partnership with Institute of Forest Genetics and Tree Breeding, Coimbatore. Dr. Yashoda was the project coordinator, and two Department of Biotechnology, Government of India programs we used to develop this database in the case of Teak. So in the case of Teak, this is the first report of the draft genome that is published in 2000. Now that we have information is available on the Teak genome, including up to the chromosome scale assembly. But this is the first report in which we published in 2018 of the Teak genome, and in this we have used both Illumina high sequencing approach as well as the paired end sequencing using nanopore. And contig assembly was generated. Then we did the gene ontology prediction using Augustus. Pathway prediction was also carried out using KEGG. And this draft genome development in Teak for the first time has generated huge amount of data, from which we were able to develop the genome-wide tools for both the illegal felling applications and timber traceability. So when it comes to the genome-wide SSR, so once the draft Teak genome is in place, and we were able to mine out using MISA, we were able to mine out more than 1 lakh SSR motifs. So from this 1 lakh, because that time we sequenced 6 genomes from different accessions, including the Nilambu Teak. So we were able to assess the in silico polymorphism in this genome-wide SSR markers. So more than 16,000 SSR markers were showing in silico polymorphism, and from which we standardized 50 polymorphic SSR primates, and of which, and this table shows the different proportions of the repeats, SSR repeats in the genome, and this is the percentage of the different SSR repeats. So, and here you can see on the right hand side, you can see the allelic patterns, SSR allelic pattern. You can make out the allele number and allele size. So these are the means by which you would be able to go for individualization of timber by employing these genome-wide SSR markers. So this we were able to achieve only because we were having the draft genome of Teak in place, that too in 2018. And then we had done a couple of projects in which like we would, so the first step, like after identifying the genome-wide SSRs from the Teak genome, then as Professor Ulfa was mentioning, like we had gone for the population genetic study. So in the population genetic structure, like in fact, I'm not going into the details of the population genetic analysis. So in which like we had selected some 18 natural populations across India till Madhya Pradesh. And then of these 18 natural populations, we sampled from each of the populations. We employed initially 25 genome-wide SSRs, and we were able to identify that there are only 3 gen ecological zones. So all these 18 populations were clustered into 3 gen ecological zones, and through this project, we were able to identify the Teak populations which has unique genotypes, which has private and adaptive. So the individual Teak trees which has the adaptive allele, so that can be targeted for the tree improvement program, and it can also serve as the seed source for the future establishment of plantation. So this is the genetic admixture pattern that we observed. So this panel, you can see the left hand side, you can see all these are the population that are sampled from Kerala, and like there is a significant amount of genetic admixturing. Similarly, we have this Tamil Nadu populations that is clustered with Karnataka populations, and we have the populations from Gujarat and Madhya Pradesh. So all these together for the 18 populations across India, only there are 3 gen ecological zones. So this is the publication that we made out of this study. And then with this information in hand, so we were able to develop, so by sampling across all the 18 populations, we were able to develop a DNA reference database using the genome-wide SSR markers from the Teak genome itself. So this is the strength, like by only after developing the DNA reference database, only you can look into the forensic applications. Without the database, you may not be in a position to go for individualization, to trace the origin of illegally logged trees, or to trace the origin of samples in the Teak supply chain. So in which we have sampled 425 individuals, and we have from 18 populations. And now that we have the reference database in place, then what we did is next we subjected this database for the STRAF analysis. STRAF analysis is the STR analysis for forensic application. So the STRAF analysis identified, because even though we have a standardized protocol for 23 SSR markers or 25 SSR markers, when it comes to illegal felling, when it comes to the log to stump comparison, you cannot employ all the 25 markers. So you need to shortlist the markers, SSR markers, which has the forensic parameter, which is the best in the forensic parameter, so that you would be able to go for individualization. So we subjected the database for the STR analysis for forensics, and we identified the SSR markers, 10 best markers based on the power of discrimination, polymorphic information content, and power of exclusion, and lower probability. So based on all these criteria, we shortlist the markers even to 5, so that you would be in a better position to go for log to stump comparison or timber traceability when it comes to the forensic applications. And we even employed all these 2 machine learning algorithms, and of this mycorrhiza was found to be the best tool when compared to the SANPOP. So this serves as a potential tool for illegal timber to identify the log to stump comparison for illegal logging, like whenever it is happening, and then for verifying the timber origin. So the genome-wide SSRs and the DNA reference database established helped us to extend the work further for forensic application. This is the paper we published in 2025, in which we have developed and we have validated the DNA reference database for forensic applications. So this is the allele genotyping pattern for SSRs, where you can see, like in each of these, you can see this is the allele number. In this particular locus, there is only 1 allele, and this is the allele size. Similarly, this particular locus have 2 alleles, and you can see the corresponding alleles. So based on this only, we are identifying, or we are going for the log to stump comparison. And when it comes to the forensic applications, whenever there is an illegal felling, you will be receiving the seized logs, through the judiciary, and then you will be having the source stump. So these are the five. In one of the cases, these are the original stumps that were located in the forest. And these are the 5 primates we had looked into, and these are the allelic pattern. And these are the seized woodlogs. Seized woodlogs, you can see like these are the different seized logs, and these are the allelic patterns. And the similar color represents the similarity matching between the log and stem. So this is the way how we are doing the log to stem comparison in illegal timber felling cases. So this SO1, you can see that this SO1 sample matches with SC1 and SC2. So these two are the logged or seized wood that is provided by the court for comparison with all these source woods. Source woods are also extracted from the crime scene, and the similar color. Similarly for SO4, you can see the SC3 and SC3 the same color. So these are the seized woods and these are the source woods, and this is how you will do the comparisons between log to stump. So next comes the chain of, so next is so whatever I explained in the earlier slide was with respect to the log to stump comparison in illegal felling. So next application for the genome-wide SSR tool is for the origin verification. So when it comes to the origin verification, we know like we can go for chain of custody validation, legality of the trade timber. You can assess, suppose if your forest source is certified, you can go for the legality of the timber or the origin of the timber, and also for origin verification. And these are the 2 main tools separate. So after the development of the DNA reference or the SSR reference database, what we did is like we did a validation with 2 of the national forest and 2 of the plantation. So from these 2 national forests, we had collected samples, and from the bark and even from the increment, using the increment bore or even core woods we had collected. And then there were 2 plantations which were clear felled that point of time, and we had collected samples from these 2 plantations, and actually we did a blind test validation for the reference database we created. So this is the assignment accuracy actually, like for the, even though we collected from all these 2 natural populations, only from PAR means the Parambiculam population, only that shows 100% accuracy. This is because of the genetic admixture pattern, because we know like the genetic profile of the populations are not unique. There is an admixture that is happening in the teak natural populations in the country because the seed sources are moving across. Similarly, for the plantations, we were not able to assign the individual timbers back to their origin, because this is because most of the plantations are established using the seed sources from multiple sources. So if and that is also proven by the population genetic study. So there is a genetic admixture as explained earlier. Like, so this is the admixture pattern. So because of the admixture pattern that are existing, DNA tools alone cannot go for timber traceability in the case of teak. So then what we did is like next we proceeded with the development of the chemical signatures. So in this case, Dr. Sandeep, who is currently the TeakNet coordinator, is assisting me with the development of the isotopic signatures. So in these instances, whenever in any of the timber tree species, whenever there is a genetic admixture, you have to go for an integrated approach where you can club your DNA tools with that of the chemical signatures. So the first one is the isotopic signatures. This we already experimented in Teak in one of the World Forest ID project. Myself and Dr. Sandeep were working together, and in which we collected Teak samples across India, and we developed isotopic signatures. So stable isotopes, we know like all the living organisms will sequester all the major elements like carbon, hydrogen, nitrogen. So most of these elements like carbon, we have 2 isotope forms, 12 and 13. Nitrogen, we have 14, 15, and oxygen, we have 18 and 19. So these stable isotopes will occur in the teak timber, like in or any timbers in a particular ratio. So the stable isotope ratio analysis depends on the geographic region in which the timber is located. So by looking into the stable isotope ratio analysis using an instrument called isotope ratio mass spectrometer, IRMS, you can actually trace the timber back to the origin. So now that we are in the, because DNA tools already we developed, we already experimented, we already validated. Now that we are in the process of developing isotopic signatures for the teak trees from across the natural populations in the country, and just to have a validation whether it can serve the purpose, whether it can trace the timber back to the origin. So like isotopic signatures, we have other chemical signatures, spectral signatures that are FTIR spectra and DART tautomers. So as DNA barcode serves as a species authentication to like DNA barcode, this can actually supplement species authentication because FTIR will actually look into the wood composition. I mean, generally the wood is composed of lignin, cellulose, and hemicellulose. It will look into the functional group and it will generate specific spectra, species-specific spectrum. Likewise, DART tautomers can create a spectral profile or a chemical fingerprint of low molecular weight metabolites. So these two can, FTIR and DART tautomers can use for species authentication. It can supplement DNA barcode whenever there is a taxonomic complexity, and isotopic signatures can be used along with the genome-wide markers for tracing the timber back to their origin. So this, as I had mentioned, like we had now that the project is just got over, we developed both DNA reference database as well as isotopic reference database for the teak trees across country as part of the World Forest ID project. So these are the few publications we made out of this research in teak, like the first was on the draft genome, and this was an exhaustive review of literature, and this is the gen ecological zonation, in which we reported 3 gen ecological zonations, even though there are multiple or 18 populations in the country, major populations in the country using genome-wide markers that is mined out from the teak genome itself. And this is the paper which reported the SSR-based individual verification system to trace illegal logging, especially, and geographic origin. In fact, in some of the populations it is been proved, but in some of the populations we have to supplement it with chemical signatures. And this is the paper that is published in one of the FAO book, in which we demonstrated the use of DNA tools for identifying the timber. So with this confidence, like over the last 15 years, we were developing the databases, and finally we established a Center for DNA Barcoding and Timber Forensics at KFRI with a vision to, as a state-of-the-art facility to deal with illegal timber theft offenses in the state. So this is the first of its kind of facility in the country, because the major challenge what we faced over the years is as Professor Yongut was correctly pointing out, the extraction of DNA from dead wood. So over the last 15 years of research, trial and error basis, we had worked out so many protocols. Now that we have modified protocols and we are going to file a patent, we are going to file a process patent for the extraction of DNA from the dead wood. Even the wood is extremely shredded, or even if it is exposed to different seasons, now that we are in a better position to get good quality DNA so that we can go for a timber forensic application. So this is the, and then whenever a timber theft offense is happening in the country, along with the investigation team, we also used to visit the court, I mean the crime scene, and then the samples will be collected and it will be submitted to court. And from the court, from the judiciary, it is coming to the facility for submitting the DNA evidence in timber theft offenses within the state. So these are the different quality assurance in place. We have standard operating procedures. This is according to the UNODC forensic timber identification guidelines we are following, and we have a chain of custody register starting from the receipt of the seized specimens till we submit the DNA testing report to the court. And we have internal quality controls in place. We have equipment calibration records, and we have a validated method of DNA tools which can be used both for timber theft identification of the species as well as for log to stump comparison. And whenever new staff is joining, like we are giving them training and capacity building programs. So when it comes to the standard operating procedures, in the first instance, like the samples will be received from the court. Whenever a timber theft offense is happening, samples are received from the court along with a forwarding. Not only from the judiciary, we are accepting currently the samples, and then we will maintain the integrity of the samples, and like as we know, like we have to maintain a chain of custody reports starting from the receipt of the sample till we submit the DNA evidence to the court. We are maintaining a chain of custody report. And once if we have the successful, then we have to go for the DNA extraction. So even though we have a standard DNA extraction protocol, even though we had made several modifications, sometimes depending upon the specimens, we may have to make additional modifications. So likewise, if we are successful in 99% of the cases, we are successful in getting good quality DNA for downstream applications, and then we will go for PCR, then PCR for the either for the generating the SSR pattern or genotyping pattern, or for generating the DNA barcode reference database. Then we will be subjecting the PCR product for the Sanger sequencing. We will be doing the bare minimum bioinformatic analysis to remove the noises so that we would be able to generate good sequences. And then finally, we will submit the evidence to the DNA testing evidence or scientific evidence to the judiciary in the timber theft offense. So these are the internal quality controls in place, like starting from the receipt, we are maintaining the integrity of the samples, and we are maintaining a chain of custody record, and then the samples are stored properly so that it preserves the integrity of the samples. And for DNA extractions, we are always running a negative control. And when it comes to the PCR amplification, we need to have a positive and both positive and negative control. And then the sequencing, then the bioinformatic pipelines, and then we submit the report. So there are internal quality control measures which ensures that the DNA evidence submitted to the is 100% authenticate or accurate. So the major stakeholders for us is the Forest Department, Police Department, and the Judiciary. And this is the way we are generating the DNA barcode. This is a DNA barcode sequence for one of the crime instances. So this is the way we are. So if it is 100% accuracy, only like similarity, only we will submit the DNA evidence to the court for species authentication in timber theft offenses. And these are the different some of the cases. And when it comes to, as Professor Ulfa was mentioning, we also created a reference database for DNA barcode for 60 timber tree species that includes 16 timber tree species that are coming from outside to the market. So this database for 60 timber trees DNA barcode database is currently with us in the facility. So, and this is the one paper we published in which we employed artificial intelligence algorithms also. So this is the way by which this database helps us to authenticate the species, timber species in timber theft offenses. So to conclude, we can say, like, as per the ITTO's 22-26 strategic action plan, legal and sustainable supply chains is vital for conservation and for increased use of tropical timber. So the LSSC is the ability actually to track and trace timber every step. So whenever the timber theft is happening, or whenever the illegal timbers are cross-border trade is illegal timber trade is happening. So if we have to track the timber through the supply chain at every step till it reaches the point of sale, or till it reaches the consumer, definitely we need to have a concerted effort. So this is whatever I had explained so far is what we developed, all the databases, the DNA barcode reference database for timber theft offenses to identify the species, or DNA reference database for log to stump comparison in illegal felling, and to an extent in timber traceability, which we developed over the last 10 to 15 years of research in the country. So likewise, we need to have, because Teak is being distributed in many other tropical countries, especially Laos, Myanmar, Indonesia. So we need to have a concerted effort across the globe so that there should be a global reference database. So only if there is a global reference database in place, we would be able to track the movement of the timbers across the borders. So if we have a national reference database that can only look into the illegal felling and the movement of illegal timber within the country, but we need to have a consortium of the experts so that we can develop a global reference database. And there is a need to harmonize the forensic standards. If we develop genome-wide reference database in one country, if we develop with few markers, and in Indonesia, if they develop with some other markers, it doesn't serve the purpose. So there should be a harmony of the forensic standards, and we have to equally strengthen the legal frameworks and the certification systems, because under MOE of Ministry of Environment and Forest, Government of India, they are actually going to the implementation of forest certificate. Hopefully it will come out in another 5 to 6 years. And then last step is, like, we need to have a capacity building program for the enforcement agency, so that they would be having an understanding about the tools in place, the advanced tools in place, which can actually trace the timber back to their origin, and they would be in a better position to tackle the illegal felling. So with this, so I would like to thank all my former directors and the present director, because this whatever I presented today is the effort of over the last 10 to 15 years of research. And I would like to thank my colleagues, Dr. Donald James, who is helping me with the genomic tools, and Dr. Sandeep, currently the TeakNet coordinator, he is helping me with developing the isotopic signatures and spectral signatures, and the different funding agencies at different point of time, like Government of India, DBT, Department of Biotechnology, Ministry of Environment, Science and Climate, Environment, Forest and Climate Change, the global funding agencies like UFRO, ITTO, and World Forest ID, and all my former and current PhD scholars who were part of this development of reference database. So with this, thank you all for the very patient listening.
Okay, thank you very much, Dr. Sumar, for your excellent presentation. So before I open the floor for comment and question, I would like to say some congratulation words. First, I would like to congratulate to Indonesia and India for successful development of the legal system BRIKSS in India and SBLK in Indonesia. And both systems have been in place, I think, since 2012 in Indonesia and 2018 in India, and very successful. And congratulations for the advanced research and implementation of the DNA and genomics research in both countries. So in Thailand, I think we just started only for research activity. So mainly supported by Fortune and Geographic by Dr. Tani, but not for real implementation. Yes, so we are just the beginner of this science and for the real implementation. So I see a lot of names. I mentioned about Dr. Yasoda, the founder of the genomics in India, and very support to the research of the Kerala forestry research. And the last one, very congratulations for the successful of the adoption and application of the DNA barcoding for COC recovery, and to test the origin of the birth, and even to use the scientific research as the evidence in the court for the judgment. Congratulations again. So now I open the floor for question and comment. Any comment or question for the floor? Please raise your hand. Oh, Kun Sapon, yes. Kun Sapon also, he had the experience in the DNA and genetics.
Thank you very much. Thank you for the speaker. So I see that the DNA technology is very improved at this era. Just one question from my own curiosity. For the legal check of Teak, you said that we have to compare the DNA from the stump, between the stump and the log, right? I just wondered that the stump of if you want to check the DNA of the stump of Teak, the stump is remain in the forest, and of course it was attacked by the fungi, bacteria, or any microorganism. So how can you make sure that the DNA that you extract from the stump is 100% the Teak tissue, not the contaminated with other DNA? Thank you.
So yeah, thank you for the question, Sapol. Like, see, unlike years, where we were using general profile DNA markers. So if you like RAPD, ISSR, so all these markers were actually developed in maybe towards the end of 90s, in 96, 97, that period. So that time it is by using universal primase. So universal primase means like it can amplify, as you said, like any amount of fungal or bacterial contamination. But see, as over years, technology has advanced, and now that we are in a better position to see this particular set of genome-wide SSR markers we have developed on the Teak genome itself. So the PCR or the primer that is being used for the amplification is very much specific to teak tree. So it won't amplify any other, even if there are other plant samples or wood samples, I mean wood tissues also, it won't amplify because the primers are that much specific to teak. So you can get rid of 100% with 100% confidence. You can get rid of all these contaminants.
So it means that the primer is specific for the teak tissue or just only.
Yeah,
Right?
right.
Okay. Thank you. Thank you.
So the answer is environmental and biological factors have very less impact for matching the side loss and origin stump in the forest to track back. Okay. Another question or comment?
Yeah.
So Dr. Tani, so I read your paper recently. It's quite similar to the Dr. Suma presentation. Please. Dr. Tani, go ahead.
Oh, thank you very much. Thank you very much, Dr. Suma. I'm very enjoying your talk. Yeah, so the first, just a comment. I also joined one project to develop the illegal logging detection system in Malaysia last time. So it's quite hard to prepare the database to detect illegal logging. That's why I want to say congratulations to develop this system. And I just have one question. For the illegal logging, so if we have this system, this becomes a good deterrence to stop the illegal logging, because all some illegal logging knows there is a system to detect illegal logging using DNA system. So in India, so now this system is informed to the people, and then this becomes a good deterrence to stop the illegal logging. How is the situation? This is my question.
Yeah, right now, the Center is the Center for DNA Barcoding and Timber Forensics we established at Kerala Forest Research Institute. And you know the rampant illegal felling that is happening in the country. So what happens is like whenever there is an illegal felling in some of the high profile cases, we may have to accompany the investigation officers to the crime scene. We should be part of the sample collection. I mean, both the seized log specimens and for the original stump that is located in the forest. And then it will be submitted to court. So right now, we have restricted our activity within the state. So the center is very soon, it's in the process of getting approval from the state as a nodal center for the state for timber forensic purposes. But the thing is like you are not in a position to see, as a country, if you take like illegal timber theft offenses are happening across all states. But we are not in a position to collect, because in some instances when the trials are happening in the court, in some instances you may have to appear in the court also. So that will be extremely difficult to represent for all the states in the concerned court. So right now we have restricted our activity, but what we like in almost all the lectures like this, what I'm proposing is like we should have a national reference database or national focal point, at least 2-3 focal points in South India, in North India, in Central India, so that we would be in a better position, because that many samples are lined up in our facility, even within a single state. So it's very difficult to take up samples across the country, and it's very difficult to appear in the court across the country. So there should be a concerted effort of the experts across the country so that we would be in a better position to combat illegal logging in the country.
And also you talked that the illegal teak log is smuggled into the India from the overseas. That's why this database should be extended to the overseas, right?
So, yeah, it shouldn't be a national reference. It should be a global database with the same tools, with the same markers. Then only you can trace the cross-country movement of the illegally or smuggled timbers across the borders. So recently there was a ruling from the Supreme Court of India that the scientists should come forward to develop such a tool. But because in one of the very high-profile cases, there was no scientific evidence. That was in sandalwood. So there was no scientific evidence before the court to prove that they have convicted the crime. So So these illegal traders are actually, they can escape the conviction process. So we should, as professionals or scientific experts, we should all come together. We should put forward this concept to some of the global funding agencies, and then we should be in a position to develop a global reference database for most, not only in teak, like for in other highly priced timber tree species also. Yeah.
So ITTO should be very important
Yes.
institute.
Yeah.
Yes.
Yes.
Thank you so much.
Yeah,
Okay, thank you.
Thank you.
Yeah, so talking about the court, so I called Dr. Turasidas last week. He said that he went to the court at the experts visit
very often,
for the verification. So any comment from Dr. Turasidas?
Yes,
so maybe
Professor.
Maybe
Professor,
DNA can help you.
Yeah, because being an wood anatomist, I use the wood anatomy as a basis for species identification. Whatever the timber which is getting imported to India, because as Suma said, there is no legal framework in place to stop illegal trade of teak or other timber from outside the country. That is it's not happening in India, because we accept all type of timber without any restrictions on its origin or its legality, because the demand and supply is very, it's not matching, because the demand is very high and the supply what we are getting from inside the country is not enough to compensate for our needs. So we getting timber imported. So as we used to appear in court, in case of most of the case it is sandalwood, or in some cases it's red sanders, which is a site listed species, IUCN listed species for restricted trade. So I appeared 3 days before in a court for authentic identification of timber, which I gave the report 15 years back, and the court, the legal system in our country is very slow, in a very slow pace. So that case was taken up only very recently. So I appeared in the court and defended for the test report which I provided. That is in the case of a sandalwood origin, sandalwood theft, to authentically identify the species. So my case was very straightforward, whether the timber which is sent by the court to the Kerala Forest Research Institute is the real sandal or some substitute or some other. So I identified it as sandalwood, and I defended before the honorable court, and they accepted my test report. So they may give verdict or the punishment they give. So in the case of red sanders also, which is being totally banned for export from India, because it is also illegally traded from the country to outside nations. So there also, I today at 4 o'clock I have online appearance before the court to defend identification of the red sander. So it is at 4 o'clock fixed today online video conference. So again, these are some sort of illegally trading happening in the country with regard to the highly priced timbers like sandal and red sanders. So as Suma's expertise in DNA barcoding and establishing a forensic timber lab with regard to its traceability is a very good initiative with regard to some of the highly priced timber trafficking, which is illegally happening in our country. So because it will take a long time to establish a database for even if it is teak from, because teak is grown in over 80 countries and traded to India from currently imported from 80 or 50 or 55 countries now. So all along its supply chain, it's very difficult to trace its origin, where it is coming from, unless we have the place of loading and the documentation in hand, the customs documentation in hand. We will be able to trace to a certain extent from where it comes, because in European Union and all, they are very strict. They will not accept any timber without any legality or traceability documentation accompanied by its export timber. So in India, it is not like that. We can import any type of timber from any country without its traceability or legal origin documentation in hand. So the country has to take initiative for the forest certification or the illegal timber trafficking checking at one certain point. So it may take time, but this sort of initiatives is good enough to establish a database for highly priced timbers. But it will have takes time, and it is a long time process to get it verified and establish a center for forensic timber in Kerala Forest Research Institute or in other places.
Okay.
Okay.
Yeah.
Thank you.
Yeah, thank you. So may I ask the last and short question or comment from the floor before we close the session today? So I don't see any hand. Okay. I think it's proof that the Kerala host a lot of experts for the DNA and also the other techniques. So before I close the session today, so I would like to recall the remarks from Jennifer that the DNA barcoding or the technology have been advanced a lot nowadays, and this technology can strengthen a lot of the legal system, such as verification of the species and the origin, decay in the harvesting permission and trading, and support the enforcement of international regulation, such as EUDR and the FSC, APSC, and Dr. Turasidat already mentioned about the trading with the European country and provide the scientific evidence for investigation into the illegal logging and timber branching. So today we learn a lot from our two female experts, Professor Sirinas from IPB, Indonesia, and Dr. Suma from KFRI from India. So both presentations are excellent. So may I invite our audience to give a big hand to both speakers for excellent presentation. And of course, I would like to thank the ITTO for deliver the opening remarks, and on audience joining the seminar today, and a lot of various comments and questions from the floor. So we still have two more webinars. So the next one will be held sometime in early October under the themes about the teak wood property and engineered wood for furniture and wood product. And the last one may be in early December for the closing of our webinar. So without further question and comment, so I would like to close the 10th webinar today. And Dr. Turasidas, Dr. Ma, and Dr. Tetra. So may I invite you to remain in this meeting room. We still have more minutes for discussion. So thank you very much and goodbye. And have a good day.
Thank you. Thank you.
Thank you.
Thank you. Yeah.
Thank you.
Thank you. Thank you. Thank you. Bye-bye.
Bye.
Thank you. Thank you. Bye bye.
ขอบคุณครับ
Thank you.